Good evening. This is Sue for the MJMA Mycological Association. This is Taxonomy Tuesday on February the 11th. The topic of today is member spine. Does anybody have any fungi they would like to start with to discuss? No one. Well, then I guess I
have
can
one
start.
from last week.
That's good, Chris. You want to share your screen?
Sure thing. Let me open my iNet real quick. Screen.
Yes, if others want to share anything, if you just type your name into the chat and we'll get to you in order.
All right. I showed this one last week. And Brandon said I could probably get some spores out of it. So I did that. And I kept that thing moist for a good 2 days because honestly, I got sidetracked. But then I got some interesting results. There's a few of them in here. That one kind of just looked like dished in and flat. But then I found weird ones.
Could you give me one more hint? Is this a triglossum or what is the genus for this?
I think it's a Xylaria.
It's Xylaria.
Yeah, the spores support the Xylariaceae. These are slits.
Yeah,
See on the spores.
they're
And
splitting. They're splitting along that.
They're cracked open and leaking out. And there was bunches of them doing it. Like basically almost everything. You could see that. They're all...
So is this a spore that's leaking out, or is there some other thing that is leaking out spores?
No, the spores are all split open like little Pac-Mans. And the contents seem to be spilling out.
And even more in support of Xylaria was that first one that you said, seemed to, it had that flat edge, right?
All dished in almost.
Yeah. So certain species of Xylaria, this is pretty typical where, from one profile, when you're looking at it from the side, it'll have a flat side, and I call it kind of coffee bean shaped. But then when you look at it from the top down,
it's like a coffee bean.
Yeah, it looks like this. Yeah.
Yeah, and I was just like, oh no, they're all but it was weird how it dropped
even more
like that.
In this photo, it's tough to make out, but I think I can make out that faint line.
Oh, right down the middle.
Right down the middle.
Yeah, I can see it too. The slit.
Yep, the germ slit that, it's tough to see if it runs pole to pole, because some of them run pole to pole. Some of them are just like in the dead center, and some of them only go down partial way. But you can see that this one isn't the twisted one, so that rules out Longipes, which has the twisted germ slit. And then the coffee bean shape with the presence of the germ slit, I think all that points to longiana. I think this thing is pretty good for Xylaria longiana.
Interesting.
So is this a different way of having spores instead of being segmented? They just break apart when they get expelled? I'm trying to figure out what I thought the smallest thing that we saw for reproduction was a spore. And now I see the spore coming apart.
They're
Yeah, the first time I saw this, and in spores that I've scoped for Xylariaceae, and which is more than just genus Xylaria, I was sort of didn't know how to explain it, and I still don't really know how to explain it. But I've seen this also, where the slit becomes a line upon a division line where the spore separates, and I don't know if any. It looks like stuff is coming out there.
Yeah, that was my thought. I mean, I gotta figure that's probably not a good sign. I mean, I've had stuff that I forgot about, and then you start seeing, you start seeing that little tail out of a germ pore or something. That does not seem to be what's going on here. Like they've gone bad or something.
I would say the contents of the spore, and maybe it's whether it's oils or air, are just escaping through that crushed germ slit.
So these could be air bubbles and water bubbles?
Potentially. I again, I'm
Okay.
with everyone else. I don't know exactly what's going on there, but yeah, maybe air bubbles or whatever a guttules is, little oil droplets. Yeah, I don't know.
Okay, that sounds more understandable. Okay, and not to interrupt you, Chris. You want to continue?
No, that was it. That's all
You're
that
only
I
on slide
had.
9 of 13.
Oh, I already skipped through the other ones
Oh, okay,
there.
you went back. Okay, right. Okay.
Chris, did you measure the spores?
Oh yeah. There you go. That's it. 400.
Wow, I don't know how your scope is calibrated. So
You know what? I went over the thing. I bet I could look for the pictures that I took. I took pictures of the micrometer slide with my things. I was gonna bring that up at one of these and maybe have somebody help me with what that really is.
okay, yeah. So that's a good idea. We should do that at some point. If you have a good
I took pictures of all the different magnifications with the
what
slide.
you need to do is make sure that you have one line on the micrometer lined up with your reticle.
Yeah, that's pretty much what I did.
Okay, well, yeah, that could be fun.
Yeah.
Help calibrate your scope.
Yeah, because I got to figure other people are going to eventually do that or want to do that.
I just checked one species of Xylaria on Michael Quebec, Xylaria phyloformis. I don't think that's what this is, but it does mention the spores containing guttules. So that's my guess. Also, is what's coming out of the opened slit.
How do you spell guttules? Guttules?
Guttules, right?
No, I don't think there's an O. I think it's Guttules. I'll check that.
Thank you.
Yeah, and yeah, my spelling is terrible. The Xylaria filiformis doesn't look good for this thing, though that is a nice note, Dave. I appreciate the note on it containing guttules, the spores containing guttules, but that thing's very thin, right?
It's not. That's not what this is. But I didn't see the species name that you had mentioned on Michael. What was the name you mentioned?
Longipes.
Oh, you think it is Longipes? Okay, because I think I know what that one is.
Not longiana. Oh no. I'm thinking longiana, not longipes.
Okay,
Sorry.
yeah, because I think I know what Longipes looks like. This looks different.
Yeah.
Yeah,
Good call.
yeah. They don't have Longiana, so I just looked at anything, just to see what.
Mushroom Expert has the Longiana on there. I
Oh, they
believe.
do. Do they have photos of the spores? They might.
I think so.
A lot of times they do.
That is not one of the selections in INAT, that's for sure.
Okay, if we want to move on, Brandon, do you want to show something next?
Yeah, sure. And I was just looking at that. Actually, I'll bring it up and share it here,
Okay.
right? Let's do that.
Yes.
Can you guys see my screen?
Yes.
Yeah.
So this is the page on for Longiana, and here's the name up here. I mean, looks pretty good. You can see that slit has that same kind of coffee bean shape to the spores. Size kind of fits. Yours looked like the reticle might have been about 5. So if you do that at, yours looked maybe 4.5 long. So looks like it's in that range.
The spores here on Mushroom Expert for Longiana, it says 10 to 13 for the length.
Yeah, when I was saying 4.5, I was talking about divisions on the reticle.
Oh, yeah. What was the magnification on that on Chris's spore photo?
400, he said, right, Chris?
Yeah.
Okay. All right.
So if it's 400, then you have to multiply by 2.5
2.25, I believe.
or 2.25.
Oh, no, 2.5. You're right. Sorry. 2.5.
Yeah. Probably right around that 10 to 13 range then.
Yeah, the photos of the fruit bodies on Mushroom Expert look like they fit what Chris has.
All right, so
I think I might have that one. Nice.
I want to start with this one, which is some kind of rust on a leaf, I believe. And Chris can probably help me. I think you know your leaves. I'm trying to figure out what this thing is, and right now I'm thinking
Is that a sharp tip?
Yeah, I'm thinking cottonwood because of that. But does cotton?
I got the tree book in front of me here.
Yeah, kind of a heart-shaped thing going on with a tip.
Hmm.
And
yeah, I'll look around for that.
Whatever this leaf is, this black ascomycete was only affecting those leaves, and I didn't get a great proof of that, but you can see probably the tip on this one too, and the heart shape. But it was only affecting those leaves. So if I can figure out what that leaf is, I might be able to figure out what this thing is. But yeah, pretty cool. And they're not just little dots, which is what you see a lot of the time. They have a little bit of character to them.
Yeah, they look like they're kind of raised.
They are. Yeah. That's what I was just about to say. They're kind of three-dimensional, right?
I have an idea on the leaf.
What do you got?
Red bud.
Red bud. Okay. Would you mind throwing in the well, I'll remember red bud. Do you have a genus for that?
I'll get you one here.
Yeah, it's fine. I can
Circus
Circus canadensis.
canadensis.
Yep.
Circus canadensis. Circus with a C or an S?
I'll type it in the chat. Hang on.
Cool. Thank you.
CER.
CER. Okay. All right. Nice.
Jason's Mike says he's not working, but he suggested a Simlax, SMILAX leaf.
Simlax. All right. And then while we're on.
Oh, Milax. Yeah. That's a good one.
You know that one?
It's sticker bushes.
Oh, really?
That's a good one, Jason. You might be on that.
There are two types of smilax, actually three, I think. One is a very round one, and one has more of a pointed tip like that.
Yeah, the
Okay.
greenbriers. Were there any of those around? It could be that.
That's a it's a cat briar.
Yeah, that's really weird because I found it at 2 locations over the past week because, you find it once, you find it again within a week. That's how these things work. The first location didn't notice any sticker bushes. In fact, it was a pretty clear area, but it wasn't like a domestic park. So maybe they clear out the sticker bushes. The second location was more, I kind of got a bushwhack back there through a ton of sticker bushes. So second location, definitely a lot of sticker bushes present. The first one, not so much, but again, maybe they, have people taking care of those there.
If you look at the where the stem of the leaf hits the leaf surface, if you got any of that, man, I think that's redbud. I think Lila's got that one.
Right here.
Yeah, it's set in, and then there's 5, 6, 7 little stems. It looks like in like some of these pictures I have, like little veins, which that has a bunch of them. The Smilax has like one main vein that goes up the leaf, and it's a hardy leaf, a stiff leaf.
So maybe I have both in here.
I suppose it's not impossible. But that one all the way to the right, I would say that is a real good fit for a redbud leaf.
All right. Redbud or sticker bush is a better start than cottonwood. So
Yes.
I appreciate that. All right.
But what about the fungus? Do we have any names for the potential fungus?
What did you see about that?
Nothing. I mean, there's a bunch of these things that without the host, it's like it's futile, but it looks very similar to a thing that I saw on Sam Bucciarelli's page. I almost just called it that, but I didn't know the host, so I'll probably go back and review that. While we're on the topic of host specificity, this is a cool thing. So, these are tiny. I thought they were a little dried up Excidia, when they get really dark and really small. But no, they're clusters of little cup fungi. And I know the host on this one because it's one of the few trees that I feel 100% confident IDing. and I'll give you a proof of that in a moment. But this is under the scope, or the stereoscope. You can see how many of these little discs are actually packed in that tiny, tiny little area. So this isn't the best photo, but you can make out the many cups that are packed within a 1, 2, 3, 4, 4.5 millimeter area. So I'm saying these things are within somewhere between 0.5 and 0.75 millimeters, each one of these little cups, which, I mean, anything less than a millimeter is just impossibly tiny, right? But defined cups as well. You can see that they're concave. They're not just flat on the surface. This is a little mystery ghost guy in the background that I'm not sure is associated with the black thing. I actually don't know if that's maybe Apothecia the lichen, but I included the photo. And here we go. This tree I know this tree very well. This is Liquidambar, right? This is a sweet gum. It can't be anything other than a sweet gum, right?
I agree with you.
Not with those birds. Yeah, it's good. Good
Okay.
idea.
So I put it up as Tympanis. I don't know how to say this. Tympanis. Which, when you're looking at these things, they're all named after the host tree. Not all of them. There's a couple of conspersas and things like that mixed in there, but it's not a very large genus. They're plant pathogens. And I was lucky to find this and be able to ID the host, because this tree came down in a storm and I think you can see up here rapidly, beavers are getting to different parts of this thing. But it came down less than 2 weeks ago, I would say. And so it still had a couple, of those seed pods or spike balls. But I haven't been able to find I haven't been able to find good descriptions of anything, any species of Tympanis. And I had definitely not come across one that grows on Liquidambar. So if anyone has a recommendation on a paper or a key for Tympanis, please let me know. Anyone found one of these before?
No, but I was gonna ask if it's like, was it on a part of a tree you could have never reached any other way?
Yes. Yeah, that's why I mentioned that.
I always wonder whether, like we're just missing a bunch of stuff.
Yeah, canopy branch.
Right.
So I still have the thing laid out for spores and to do microscopy, but I haven't been able to find a good description for anything in this genus. So, what's the point, right? Until I can find something to compare it to.
Tell you a story 600.
Do you know what family Tympanis is in? I don't know anything about this genus.
So I think it's a Perino.
Let me tell you story 800. A titled Greek girl insists on burying her rebel brother and gets entombed in a cave. The gods
No, that's it. Yeah. Llyophyllum.
Oh boy. Tympaniaceae.
Yeah, Tympaniaceae.
Look at all the species.
Well, the species, and look what they're named after, right? Abietina.
Yeah. Is this thing, was it hard like something in Xylariaceae?
Yeah. Well, it's tough to say, right? Because I found these things, some like with Orbilia, right? I think it probably is in that. Actually, I don't know, because it's so small. Unless it's like actively rehydrated, which I did, and I tried to figure out if it was, hard to the touch, or had a bit of a, like a Scutolinia type feel to it, where it's more jelly-like, but they're so small that it just feels like a hard little thing between your fingers, right?
Yeah, and plus at this time of the year, you might not get an accurate representation of some aspects of these things that sort of hang around for a long time.
Correct. Yeah. But let's see what else is in Tympaceae. Tympanaceae. Bunch of things that I don't know what they are. Yeah, cool thing.
Yeah,
Glad
a whole bunch of names I've never seen.
Yeah, right. So that's my weekly. Here's a cool thing, and I do know, I do believe it's Tympanis, because I've been trying to figure out a bunch of other little black disc type things, and I keep coming across this name. I'm like, no, none of that looks good. No, none of that looks good. And then I saw this, and I was like, oh, I know exactly what that thing is, because I've been comparing it to a bunch of things over the past couple weeks. And then Nick came in, who he knows these things. He came in and confirmed Tympanis, but the problem with INAT, like I asked him, okay, you got a key, you got a a paper that I can read to compare, and
Would
didn't hear
Seaver
anything.
cover these things? He's got a 2-volume thing about cup fungi. Is this still in the cup? But you said it was in the Pyramitis? Pyramites?
That was a guess. Pyrene, a pyromycete type thing. Doesn't look like that's the case, judging
Okay.
just by what we looked at.
Maybe the Seaver book has something.
Yeah, so maybe it's a disc in that cup fungi paper. 735. Yeah, let's look at this thing, and then I'll let it over to the next person. So I found this today. It's laid out for a spore drop. I did a quick micro on the spores. I didn't upload the photos. The spores are subglobose to broadly ellipsoid. I don't think this is one of the kind of cold weather Mycena type deals. I think this is a white spored thing, but I don't think it's Mycena. My first thought is Llyophyllum. And it's interesting because I found it in a place I go all the time. You guys have heard me talk about Black Roan Preserve. It's a recently burned preserve, burned last spring. Mixed woods, conifers. This was under holly, oak, and pine. But yeah, also in very much burned soil. I don't know if you can really make that out from the photo, but just messing with the soil stained my fingers. Here you can see them over here, over here. But yeah, I only uploaded these 3 photos just recently. The other thing is you can kind of see that, I don't know how to translucent quality to the stipe, where it's got that, kind of shimmering thing going down the stem. But yeah, anyone have a guess here, or think
the gills look a little bit yellowish.
A little bit?
Yeah, I genus Tubaria, the species in that genus tend to fruit in cold weather.
Yeah. Yeah, Tubaria is definitely not a bad thing to look at for this one.
Yeah, Tubaria have most of them have like yellow to ochre spore prints. Of course, if you don't get enough spores to drop, that's hard to assess. The spores of Tubaria are typically not really big. They're smooth. Mostly they have one gutule, and they're a little bit asymmetric usually. Kind of the walls collapse on them, so they look a little weird.
You think a Tubaria would have this color cap?
It's not. I don't think it would rule it out. Tubaria furfuracea is considered to be a complex of species.
Yep.
And then there's a few other tubaria also. But I don't. This was in habitat that I'm not familiar with. And you do find these weird things in these burn sites that you don't find anyplace else. So.
Yeah, and there's one, like I said, I found it at probably 3 this afternoon, laid it out for a spore print, got one clear, like 2 millimeter by 2 millimeter, where you could see the striations from the gills, but you couldn't even see like the color. I don't know if anyone knows what I'm talking about, but if you hold it up to a light, you're like, all right, there's spores there. Can't make out the color.
See if you can. If it's on a slide, put it on a white surface and see if you can notice any sort of like pigment.
Yeah, I already looked at those spores.
Yeah.
And they weren't, to be fair, they weren't tubaria spores. They were very much subglobose, pretty much round.
Okay. Well, that would kind of align better with Llyophyllum as you suggested to begin with.
And just a real quick, doing real quick research, there is a burn associated Llyophyllum. The pictures didn't look great, and I need to do some micro to confirm, do some spore measurements and things like that. But it's a possibility. Figured I'd share it.
Yeah, with tubaria, you can usually find Chylocystidia pretty easily, also, and they're like these like wavy, cylindrical things.
Okay, they're not. Would you call them filiform?
No, not quite. No, they're a little. They're a little thicker filiform.
All right, cool.
I think now there might be a tubaria that's like that, but I'm used to looking at tubaria porphyracea because a lot of times that's the only thing I find in the winter. So I bring them home, and there's nothing else to look at. So I do some scoping on them, and these don't really look like tubaria porphyracea, though. To be honest, it's just all I'm noticing is that it looks like the gills are slightly pigmented, and it's the right time of year.
The gills were the thing that were kind of holding me back from the gill color specifically, because they are very much yellow. What's the other thing that fit? The stem is hollow, very much hollow, not pithy or anything like that. So just another thing to compare.
Yeah, there's all these weird genera also that have only like 1 or 2 species in them, like Gemundia.
Yeah.
I don't know how to ID these things really. So,
cool.
But that's you got some material here that's in pretty good condition, and you can dry some out, and you can do a little microscopy, and so that's a pretty cool thing to find. Whatever it is.
Yeah, man. The actual mushroom growing on the ground this time of year is fun. So, all right, that'll do it for me. Thanks for letting me share, guys.
Thank you, Brandon. Lila, you're up next if you're ready. Lila, are you around? Okay, she's starting her screen share.
Okay, got off mute. That helps. All right, so this was, I don't know, a little over a week ago, probably, out tromping around in a swampy area through some of the snow and areas that I can't go when the ground isn't frozen, basically. And I thought this was kind of cool because it's exhibiting geotropism. Not the best photos, but you can see right here in the center that this fruit body, when the tree was standing, was growing this way, and now that it's down there, they're growing off to the side. So let's see, I gotta get rid
Yeah,
of
it looks
this.
like the tree was standing and then it was leaning, and finally it came down, and it looks like they were growing at these various stages of the stature of the tree. It's interesting.
Yeah. Yeah, so it's had some evolution on it. Yeah, here's another one over on the far right, going at 90 degrees roughly. So anyway, here I zoomed in on that older one from when it was standing, and these are obviously old too, but I thought it was cool to see that. It's some sort of a Ganoderma, maybe Applanatum. You can see I don't know if it's seasonality or 2 years worth of growth or what, but you can see the tube layer, 2 different phases of growth anyway in it. And a little close up on the pore surface, not the best focus. But yeah, so anyway, that was something, and it caught my eye with the geotropism, so I thought I'd bring that to show.
So what do you think that is? I see the pinkish on that, so I always think Compragosa species, but
Yeah, I don't think it's Compragosa. I think it's,
because the top doesn't look that way.
yeah, I think it's a Ganoderma, but it's kind of too far gone to really say, like, doing the thumbprint thing in the top wasn't valid.
That's interesting that it's still stained though when bruised. A lot of times the Ganoderma, once it's winter and they're no longer, the pore surface is no longer viable and active, you get no bruising on it. Also, those pores look maybe a little bit large for Ganoderma.
Kind of big, yeah. Yeah.
And it's too
I thought
small
the same
for
thing.
Dilopsis, so kind of interesting.
How can we be stumped on something that's
So
looks
common.
yeah.
I know.
Oh
Well, it's
God,
just-
old polypores.
Yeah.
They're very frustrating.
That's
to
the
me.
problem,
Yeah.
the age of it. But
Because you might have there might be secondary growth on this as well. And I don't mean like another seasonal growth. They're,
there's some mold or something on it, yeah. Yeah. So, but it has that Ganoderma kind of look to me, but that was that one. What else? Here we go. Here's the Llyopsis Compragosa.
Yeah, it's too elongated. Yours
Yeah.
is a completely rounded, yes.
And it's a thinner, this is a thinner polypore overall than that other chunky thing. And they have these
And zoning.
zoning, very distinctive. Also, gets that bruising kind of effect, but you can see the very thin walls of the elongated pores. So that's what I think that one is. I didn't write down what it was growing on, but it was on a downed log. And then this one, Fuscoporia gilva. I always like to find this one. It's, you see all these little tiny things growing on dead trees, and they're usually Trametes or something, but immediately this isn't the ones you see all the time. I don't find this very often, but usually when I do find it, it's there'll be a fair cluster of it on the tree, and it just has this different color cap. Again, it's old, but it was obviously something different that stood out, so I went over to check it out. And you break it open and you see the golden interior. mustard, I guess some people call it mustard, color. So it was just a nice contrast of color to see. So I took a minute and took a couple photos.
Is this one of these dyer friendly things?
I don't think so. I looked to see if I could find any information on it. Is because, yeah, you think, oh wow, maybe it makes this nice golden color, but I didn't find anything on it.
Hmm.
I looked in the mushroom color atlas, and I didn't see anything about it. So, but it was pretty. It was nice to see the contrast on yeah. There's some ice crystal on it. And again, it was in a spot that I can't usually get to because it's either too swampy or infested with too much poison ivy to wade through. So,
Is that a dead ash tree there?
yes, it is a dead ash tree, which is one of the hosts. I don't think it's the preferred host for this, but I believe I did find somewhere that it grows on quite a few things, including ash. So there's a lot of dead ash in this area. But again, in an area not generally accessible. So that's it. As to those three, I think. Did I show them all three? Yeah, I showed all three. Yeah.
Thank you, Lila.
Yep.
Paul, you're next. If you have something and you're ready.
Yes, I am. Let me just figure out how to share. I always have to relearn this. Let's see. Here, yeah. Here I got inspired. It was about a month ago, I think Lila showed us this Diatrype, this Kiwi Bubbles.
Oh, that's great.
Yeah, this one, so this was actually, I think about 3 weeks ago, but I went out, decided to go out looking for it because I understand it grows only on beech. And
Can you
there's plenty
share?
of beech trees around where I live. So
Paul,
lots,
can you
and
share?
I'm sorry, am I not sharing? Gosh.
Not yet.
Share. What's going on?
It's starting.
Okay, now we're going. Sorry about that. All right. There we go.
Yes.
Is it? So you're seeing it?
Yes, we are now.
Okay,
Thank you,
good.
Paul.
Thanks.
Lila.
Thanks, Lila.
Yeah.
Yeah, I think Lila, I think about a month ago you showed this one. And so again, I got inspired to go out looking for it. And indeed, it didn't take me long to find it. In fact, if this was growing on a small beech tree that was this branch, was still attached to it, but it was a dead twig there, and it only took me about 15 minutes to find it. There's a better close up of it.
Yeah, so tiny. Yeah, great. But those
Yeah,
beautiful yellow and green, nice.
I know it is. It's really you know it when you see it, obviously, but you can kind of see it erupting out of the surface of the branch here. And although I did try to get some spores to drop off of this, and I left it on a slide for about a couple days. But I think this is a little early, perhaps maybe for it to drop spores. So I did get a microscope quite recently, finally, and so what I did was I decided to kind of just take one of these growths and just scratch it onto a slide and create kind of a debris field of this material there, and I was able to find some spores for sure. There they are. Rather, I guess you would call these perhaps cylindric and maybe allantoid, cylindric, I suppose. But there was quite a few of them that ended up on the slide. It's an ascomycete. And it's interesting because some of the spores sort of stay clung together, you know, in these clusters here. That here's another shot of, and I think these are individual asci that are just these very tight clusters of these spores that are grouped here in these little packets. There was another one where they're now getting a little more spread out. I guess these are getting closer to maturity and getting ready to be expelled, I suppose. But I think they're immature. At least I did get some measurements on them, and they don't seem to come quite to the length. They're supposed to be 12 to 14 micrometers long. I did get some measurements of some that really only, it's just a small sample, but I only got them at around 10.5 micrometers long. So I have reason to believe that perhaps they're not mature, which would make sense considering they didn't, you know, drop off of the growth there onto the slide. Perhaps they, you know, I know they turn black and get, you know, more withered looking, and perhaps that's when they start dropping spores. But anyway, it's fun to find something like this, especially when you have a new toy like a microscope, and was able to actually see something interesting this time of year where there's very little to see. But I would encourage you, if you do have beech trees around your area, to take a look. It's pretty easy to see the beech trees this time of year because they do hold on to their leaves, and they're all over the place here. And I went out 4 times looking for these, and 2 out of the 4 times I did find them, and I did make an observation into INAT on those. But that is, like I say, that was about 3 weeks ago. But that is that's my only one. I don't know if anyone else has gone looking for this one.
Yes, haven't found one yet.
Chris, did you find some?
No, not yet.
Okay, I looked. We were out for a walk about a month or so ago where there were a lot of beech trees. I didn't see any. The woods are really not very nice right now. Everything's coated with ice around here from the sleet storm. But I will say about spore dimensions on ascos do tend to vary somewhat with maturity. The immature spores are often either not as ornamented as the mature versions, and sometimes smaller. So I think you're on target here, Paul. And I imagine you took into account the curvature of those spores also when you estimated lengths.
Yeah, my program here, I can actually kind of create sort of a curve, not a curve, but at least straight segments.
I see.
You know, cut the lengths there. So again, it's a small sample.
These spores are very thin. So if your second dimension, your width is very small, looks like these spores are probably only maybe less than 2 microns wide.
2.2.
2.2. Okay. Yeah, that's very, very narrow. So, you know, if that fits the description, then but this, I mean, it's pretty clearly what you have here are these.
Yeah, I don't think there's any mistake really is what we're looking at. But anyway, I just it was fun to go out and find something that's fruiting or fruiting this time of year. So that's really the only thing I've got to show. So
Nice find, and Paul, that's good work on the micro for.
oh, thank you very much. Yeah, it's fun. I got a pretty modest, microscope, and but it's fun to play with. I'm looking for things to look at, and I got a moldy orange yesterday, so I was looking at a.
One thing I want to know, what program are you using there for the spore measurements?
This is called Pixie Meter.
Yeah. So when you were the immaturity of the spores is absolutely on the table for them not really matching,
Yeah.
not being large to match Diatrype virescens. But if this is the program that I'm remembering, it takes you have to input certain information to it, right? So you got to make sure
Absolutely.
that information is 100, like really right on.
Yeah,
Because you can throw
yeah,
off
that
measurements easily.
I'm sure. Yeah, once you're down at these sorts of measurements, yeah. I mean, I suppose I can give it another calibration and see what it turns out. But that's true. I really only calibrated it the one time with my slide there. So
Could we see those bunches again?
oh yeah, like I say, this is where they're very, very, very tight little clusters here.
Yeah,
Almost like look.
it's a true sac fungus, right?
Oh,
So sometimes they call all ascomycetes sac fungi, but there are certain asci that really are, like just a sac, right? This little bundle.
yeah,
I saw this
right.
and I just cannot help. And then the spores too. It is very much like a bunch of bananas.
Yeah, yeah.
Yeah, it does. Yeah. And again, look, depending on where you're looking, this I think might have been prepared on a different slide, but and again, you see these clusters where I almost get the feeling that these spores just are, continue to cling to each other for a certain period of time after, or maybe it's just how I just kind of manhandled this slide preparation here that, but I'm not sure whether some of these spores are really contained in anything or whether they're just, clinging to each other before they, reach final maturity.
Yeah, that's great. That matches what you find online. So I'm psyched. I
Yeah,
was wondering if anybody had found any, and
right.
you shared. Thanks.
That
I
was
Yeah.
just
a
checked.
really really nice photo of the fruit bodies. Was that taken through a loop or something, or some other magnifying device?
No, no, this is actually my old iPhone. But I put on a different camera app than what comes native to the phone. This one I can manually focus, and again, given good light, most phone cameras can do a pretty darn good job if you can just control the exact focal point. So this is an old iPhone.
Okay, I just did a quick search on iNaturalist. We're up to 1,017 observations of the Kiwi bubbles.
Okay.
And since January, from January and February, they've had 485. So half of the observations came this year.
Interesting.
And of course, I don't know when that email first came out to tell everybody about this and whether we were the first to receive it or not.
Right.
That's interesting that we were able to double it just by sending that out.
I know. I'll have to take a look again.
Yeah, the guy, I'm a songster who was kind of driving this. He sent it out to a lot of clubs, so there's probably people all over looking for it. But I thought this was a fun one.
Yes, absolutely.
I wonder if the spores mature when it starts to get a little warmer.
Yeah, good question. Yeah, I'll have to go out go out and look for some more samples and try to look for ones that are a little more advanced and see what I can see.
Thank you, Paul.
Oh, thank you.
Yeah, and just on a note on those things, I mean, I've spent a lot of time around beech trees. I look for it often. I've only ever found it one time. You know, I've been aware of the thing for about a year now, and I look for it on all the beech sticks. I've only ever found it once. So it's cool you found it twice over the last couple of weeks.
Yeah, maybe I'm just lucky, but the first time I went out, it was literally 15 minutes and I'd already found it. So maybe it's, I think this fellow, Amos Oxter, I think he's in the DC area, which is sort of close to where I live here. And so maybe it's coming up from the south.
Yeah, maybe it's a more southern thing, or doesn't like our particular, coastal, at least maybe here in South Jersey, coastal plains.
Did Washington have a drought this year?
Oh yeah, we well, yeah, I live in Baltimore, but yeah, it was some really bad periods here. I imagine it's kind of similar to what you had in Jersey there. It was
Okay, so not that
bad.
different then. Okay, it's 8 o'clock. Dave, do you want to start going through some of yours?
Yeah, sure. I've got old stuff, but it's old stuff that's been updated because we had some sequences come in, and I think one or two of these things are from New Jersey. So I just sent some emails, and we can just quickly go through the Mushroom Observer posts.
Are you able to see my screen?
Yep, can see it.
Okay.
So
Right.
I found this in Cedar Swamp. It's a park in New Jersey. I was there in 2021, I believe. The name of the park is listed. Might have been Wells Mills. I'm not sure.
It
Yeah,
is
Wells Mills County Park. Yep. And I thought these were Hypholoma because it was in a sort of a habitat where you find some of these small Hyphollomas, and it looked a little like Hypholoma to me. And it's fruiting from some sort of coniferous cone. And I got some spores, and I got a spore print too. And the spore print, I should have sort of tipped me off that Hypholoma was probably not right, because the spore print is more cigar brown than dark purplish, brown, almost black. But anyway, these got sent to Steve Russell, and I'm not sure if I sent this, or maybe this was passed along to Igor, because it was at a New Jersey NJMA foray, and Igor may have submitted this. I'm not sure. But anyway, we got
it might have been. Sorry to interrupt. Just to answer, that might have been included in the blast that the club did for fungi that we found at forays.
Yeah, probably. That's probably how it got in. They're nice fruit bodies, though. They're nice. I mean, you can see a lot of detail on them. And we got, it's this Pholiota that I never heard of before. I forget the name of it already. It's here. Pholiota stratosa. Now, Nina has one also found in New Jersey that was sequenced. So she's the same species. And I looked on INAT. There are no observations of this species from New Jersey on INAT. And there are exactly 2 on Mushroom Observer, this one and Nina's. And that's it. So it was a good one to put in.
Hey Dave, you remember that name? Oh God, what is it? Pholiota sphagnicola.
Yeah, I thought I found it a few months ago. Right.
Mm-hmm. Man, it looks just like that. In my opinion, but it
does look like that. Yeah,
that's cool. So
yeah, and this was in a mossy, wet area too. So I'll have to reconsider that. That I think I might have sent that to Steve also. Well, I didn't send this to Steve. This was sent by the club, but I think I sent that thing I found to Steve, and I'm not sure I've got any information back on it yet.
Yeah,
I'm gonna
I
have to
just think it's
compare.
cool, especially the cap. If you can go back to that cap shot
Yeah.
of that kind
Yeah,
of
flat scales, sort of.
flat. Yeah, look at that.
Yeah, yeah, you're right.
Cool.
I'm gonna have to compare with that
Well,
observation. Mhm.
immediately I was like, oh, I need to look at this name for the thing that I called Sphagnicola because you know what I
Right.
mean?
Yeah. Well, at least if I, if the thing I called Sphagnicola is the same as this, at least I got the genus right that time. So,
Yeah, and I sent one of those Sphagnicola things to Fundus recently. So hopefully we figure out maybe it's this thing. Huh, who knows?
yeah, could be. Yep, yep. I'm gonna have to do a comparison. All right. Well, that's that one. Pretty cool from New Jersey.
Yeah, I have a question. Not much so much a question as a comment. I was looking at this name and I'm like, well, how they propose this name? And I went to GenBank and I typed in Pholiota stratosa, and there's only one sequence of this collection, and apparently that's the type collection by Hessler from 1961. So it appears that the ID is correct, as the ITS must have matched the type collection, but there's no sequence in Mushroom Observer. So you should ask Steve to post it.
Yeah, he'll put it up eventually.
Yeah, I know you will. Yeah. Mhm.
Yeah, he always does.
It
He's
takes some
really
time
good
though,
with that.
and he's very busy, obviously. So,
He's very busy. He's very good at getting that done.
but
If
I'm just trying
it's
to
like
say,
2 weeks from now and I don't see it, I'll ask him.
right? I'm pretty sure that that's probably the name because how else would they know that this is Stratosa? It's
Oh,
so
absolutely. How?
it's an
Yeah,
obscure name.
absolutely. Yeah, it's a
Never
very
heard of
very
it before.
uncommon.
No,
Never heard
same
of it
here.
before. Yeah, cool.
Yeah, never heard of it before. Same here. Yep.
Yeah, collected in Michigan, Sheboygan County, October 14th, 1961.
Well, that makes this even more cool.
Yeah.
Yeah.
That Michigan Jersey connection, it always makes me think, and it's very cool.
Thank you, Dave. Next.
Sure. Yeah, I thought we didn't all enjoy that one. And I think this thing is something that's also now been found that's been ID'd from New Jersey, although where does mine from? This isn't also from New Jersey, is it? It might be.
No,
No.
Pennsylvania, Lucerne
Oh,
County.
it's from where I used to work. Right by my office, probably, I guess. Yeah, it's at Cortinarius that I guess Steve must have sequenced this one. The fruit bodies are nice. The observation's pretty good. And according to the two women from Arizona, Terry and I forget the other one's name.
Donna.
What is it? I'm sorry.
Donna.
Donna. Yes, Donna. Terry and Donna. Yeah, this thing Steve is giving a coded name to is the same exact thing as this Fusco. What is it? Fusco something.
Fuscoflexipes.
Fuscoflexipes, right? It's funny because this doesn't look like Cortinarius flexipes to me at all. I know Cortinarius flexipes, and there's a few things that look like flexipes as well that are a little hard to separate, but this actually doesn't look like them. So, kind of interesting. But another,
What
yes,
do we
on a
do when we
slope
have
with planted hornbeam. Yep.
What are we doing when we have a name like this temporary IN 138? You didn't change the observation name suggested on Mushroom Observer from Fuscoflexipes to that.
Well, Fuscoflexipes was proposed by somebody else. It might have been Terry and
okay.
or Donna, and then I just I did a little research and followed up on what she had said, and what she said made sense. So I looked at a blast. I think the sequence is on here. So I was able to blast it and
Where
see
would
what.
I find the sequence? Because I don't see it in the
It's over
comments.
in the notes section. So it's
I'm
over
not
like,
getting any notes coming up here.
yeah, it's over
You
on
are
the
now
right.
logged in, and that's why you don't see the information because you have to be logged in into MO.
Oh, okay. Yeah, you need to be logged in. Yeah, that's right. I believe there's a sequence available for this.
They're
Right,
usually posted in the upper
right,
right corner.
right in the observation. Hmm.
Okay.
Our
This is
fuscoflexipes was found in the Pine Barrens, and I see now, that was an observation by Nina, and I just went there, and Dave, you left a comment there at the end of January, which I just saw, which I didn't see before, and I'll have to investigate that. You were just asking questions about some inconsistencies between this sequence and another sequence that came from
No, it's a
paper.
habitat. No, the habitat. Nina's comes from the Pine Barrens, I think,
Yes, correct. Yeah.
yeah, and this comes from like this well-drained slope by planted hornbeam. It's just the habitat is so different that I thought that was interesting.
Yeah, I claim here that I ran the blast on Nina's sequence, or it came from Steve, obviously. And I'll list some papers here that I did a research, in GenBank, and I read the paper, and it seems like that was the logical conclusion. So I have to take a look at what their sequence is and do more work because now it's getting interesting.
Yeah, I'm
But
making
again,
more work for you because I think the other things that I've shared here also are based on sequences that were posted.
yeah,
Oh,
you
did
know
you
what?
see the Gyroporus,
Yeah, I
Castaneus
saw
thing?
Gyroporus, and the trouble is that the paper that um, brought those two names, Smithia and Borealis, and everybody's attention, that paper relies on loci other than ITS. So there were no ITS sequences produced from those type or lectotype collections. And everybody's sequencing ITS, so now we cannot really compare these things directly because we need a reference point, and there's
That's what I
none.
thought you were going to say. I think there's one ITS sequence on GenBank for Smithii and one for that's called Borealis.
Yeah, but that's still hearsay because people
Uh-huh.
propose these names, but they
Uh-huh.
you've got to go to the source, and the source is the
Uh-huh.
type.
That's right.
So
Okay.
again,
That's
the caveat is, for anybody who is doing, research in GenBank and who is not well versed in all the intricacies, do not
right.
believe what you see. You have to check the references. You
Well,
have to
well,
see where the
actually,
voucher came from.
I sent that observation we're talking about now, which is not this one. I sent that one as an afterthought to Sue, and I put right in the email. If Igor is here tonight, it will be interesting to hear what he has to say about this. And we just heard it.
Yeah, we just heard
And,
it, and also note that this is Castaneus 04, so there must be 01 or 2 and 03. So we're dealing with more than just two species. So
that's what it
yeah,
looks like. Yep, that's what it
if
looks like.
if you can claim that Borealis and Smithii are one of those Castanuses, then what are the other two? And are they real? Because then you have to look at the sequences and compare them. I mean, I do this all the time. Every time somebody proposes a name based on DNA, I go, and blast a sequence and try to figure whether it makes sense or not, which is a
Well,
lot of work
I
sometimes.
do that too, Igor, but you know a lot more about what to look for than I do. So I'm always interested in hearing what you have to say about this sort of
Yeah,
thing.
I'm also the skeptic, and that's why I do this, stuff, and it just
By
drags
the
on.
way, the spores are kind of large for Castaneus group on these. A few of the spores appear to slightly exceed 12 microns in length. It's a little bit long for the Castaneus group.
Yeah, but I think one you call them castaneas because they look like castaneas, but all
Sure.
giraffes
Well,
look
yeah,
like
sure. Absolutely. Yeah.
most of these giraffes they look about the same. It's just a variation, the same theme. With exception of what is it, the C.enelensis or whatever that thing is. Yeah,
Cyanescence and purpureum. Is it purpureum or purpureus? I forget.
Gyroporus purpureus. Yeah,
Purpureus. Okay, yeah,
yes,
that one's kind of different too. There's also one that's really pale. It's almost white, and it's pretty rare. I forget.
Yeah,
It's
I
named
think
Pholiota or something. I forget.
yeah, I think purpureus sequences exist, and we found one in the Pine Barrens a couple of years ago, which I preserved, and I'm yet to sequence it. So there's there's a lot of stuff uh, to get to, unfortunately, and
And
yeah,
this is whatever this thing is, which appears to be perhaps an unnamed species of Gyroporus. Um, it's very common in this place where I go in Ricketts Glen. It's always there all through the summer. It's starting in like July all the way up through October. You see it all the
well,
time.
how do you know it's the same species unless you see this?
Well, it's always, I mean, we're talking like, really small area and they look the same. They all look the same. But,
Okay, if
I
it's the
know
same,
I'm making some assumptions. I realize that.
yeah, I mean, if it's the same area, then it's possible they're the same thing. But
It's really the same area. It's this one trail on the side of the trail all the time. And it's really, we're talking like, over a distance of, maybe 20 or 30 meters. And they're always in that area.
yeah, so here's a suggestion. It is possible to gather a few of these Castaneus type things and get TEF1 or RPB2 gene sequence for them, such that we can then refer to that paper by Navid Davoudi and then maybe put names on these things.
Oh,
So
okay.
then we can, yeah.
I've got collections of these from a few different areas, these Castaneus things. They're
Yeah,
very common where I live. They grow on my property and by hickory.
right. So my point is, if we sequence these mysterious Castaneus types with TEF or RPB, then we go to the paper, we can maybe put names and then link the ITS sequences. So we know that this ITS corresponds to this TEF1 or RPB2 and that name in particular. So that's the logical, sequence of events. Because without the link, we cannot really match anything. We need something else. We need another piece of data. And
Yeah, as you're saying, there's, some hearsay in terms of like some of these names that you see on GenBank. But the one thing I will say is that when you look at the blasts because I blasted a few of the other sequences that are on GenBank for these type things, and you see a lot of different Gyroporus, a lot.
it disappeared, Dave. Can anybody hear anything?
I don't hear anything from Dave.
Yeah, Dave just disappeared. I'm not sure if he's still here. I see his icon, but I cannot hear him.
And his mic is on, but it's not no sound.
I think his internet must have died or something like that.
Okay, well, maybe he'll get back on.
If Dave can still hear us, maybe log out and come back in.
Yeah, so just to conclude this, I'm just saying that it'll be easier for us to gather a few of these Castaneus collections and maybe sequence them for those single protein coding gene loci, as opposed to Steve doing the same thing, because I think Steve is pretty much engrossed with ITS and
So
nanopore sequencing.
where would we send those, Igor?
Oh, there's an outfit in Oregon called Molecular Solutions. I think we spoke about this. Yeah.
Yeah.
So
Okay. Okay, cool.
Yeah, he's going to do the work. The guy, what's his name? Mike Gordon or something like that. I know his last name is Gordon. First name I don't remember. Yeah. So
we know one though.
He's coming back or not?
we have a solution for this.
No. Nothing from Dave.
And as for fuscoflexipes, Cortinarius, we need to do some fact checking.
Yeah, I wanted to ask about that because the first time I ever met Dave, actually, we found a thing uh, that we were calling Cortinarius flexipes that looks wildly different than these, right? Flexipes kind of has like a bluish thing going on. Uh,
I don't know that species at all. As a matter of fact, I know nothing about Cortinarius. So,
oh, yeah.
but
Yeah,
flexipes
because
means flexible stipe.
Cortinarius flexipes.
Yes,
Uh,
flexipes. Yeah, well, that means flexible stipe. Fuscoflexipes means smoky flexible stipe,
I guess so. Yeah.
or something like that. Yeah, or dark. Fusco is dark, or smoky. I forget what that means.
Yeah, I think Fusco is smoky.
Is the
So
is
I can vouch for that Pine Barrens collection that I called Fuscoflexipes because I did research in that sequence and used the paper by reputable people who are in the business of sequencing Cortinarius to arrive at that name. So I have to see the sequence for Dave's collection to make the determination. If it matches that of Nina's, then it's a close case. If not,
you,
then who knows what this thing is.
you said flexipes means flexi stipe is the peas
Peas
somehow
means foot. Yeah, foot as leg or stipe. Yeah.
foot. Okay.
Yeah. Yeah. Foot more than a leg, but maybe I think they probably were referring to the whole stipe as opposed to its base.
Agreed. Yeah, I think so as well. You were.
Yeah. Do we have Dave back or he's still MIA?
MIA.
MIA. Yeah.
Does anybody have anything else to share? Well, we see if he comes back.
His internet probably crashed out.
Yeah. Does that happen often to him?
I know his connection is not reliable because he lives in a fairly remote area, and he also lives in a recessed area, like in a sink.
Yeah.
So if cell phone, there's no cell phone signal at Dave's place. You have to climb a mountain, a hill on his property to get some kind of a signal. But if you're physically in his house, there's like no signal at all, none,
Okay.
zilch. So my suspicion is that his internet suffers from, outages just because of his geographical location sometimes.
Might be snow in there like it is here though.
That's true.
Oh yeah, still coming down. I haven't
Oh
even
yeah.
No snow in the north,
Uh,
Jersey.
I think we got probably an inch or so already, as far as I can see.
I'll just talk about that Carolinensis, right? That
Yeah, let's talk about
Okay.
this one.
there. Carolinus. What
Corallinus,
is
yeah.
it? Corallinus? Yeah. That's a cool name to put on it, and you can see why it's put on it, right? That kind of pinkish, reddish thing. It's not your true Cinnabarinus.
Well, I think it's arguable that they pretty much look the same, but there are differences between the species, morphological differences. I think Corallinus is pretty much identified from the shape of its gills, which are more blunted and not as sharp and well defined, and also the whitish blotches on the stipe, which you can see here.
But we
And
don't
that's
really
according
have
to the paper
gills,
by
right? We have ridges.
Sweeney.
Yeah, that all that I remember Rachel talking about.
Yeah. And then Steve now claims that he can pretty much identify all the chanterelles from the ITS2 region of ITS locus. So you don't have to get TEF or RPB as claimed by Rachel Sweeney. You can deal strictly with ITS, but sequencing of chanterelles with ITS is challenging. You have to use special primers for that. And I think that's been pretty much the challenge. Not so much trying to identify or make sense of these sequences as to getting them in the first place.
But
So
Were
Yeah,
you
go ahead.
saying that this one reacts or has positive results from just ITS?
No, all no. Steve claims that he can recognize all of the taxa that have been erected recently through DNA sequencing, specifically through the RPB and TEF1, loci, the single copy protein coding genes. He can
Okay.
basically say, I can take ITS2 and arrive at the same name from that sequence.
Cool.
Now, ITS1 is either difficult to get or it's just very variable, just like in Lexinum. There are some nuances in there that are sort of, it's not a very good fingerprint. But apparently ITS2, which is a little bit more conserved, is still a good enough fingerprint to get these species figured out just with ITS alone. That's what he said, to me in an email, or I should say, comment on a Mo maybe a couple of weeks ago.
And so it seems like ITS1 is a good, broad strokes thing, and then ITS2 is also easily isolated. But when you get to
Well, no.
RTB and some of these other loci, they're more difficult to prepare, or
Well,
why is
here's the thing: getting ITS from Cantharellus-like species was just historically difficult, and I don't know all the details, but it had to do with primers, and that's why people were probably looking for a different barcode, and they figured out that these single coding protein coding genes were easier to obtain than ITS, whereas historically the reverse is true, that RPB and TEF and ATP6 and other things like that, because there's only a single copy of that gene in the entire organism, okay? There's much less of that DNA present in tissue, and it's more challenging to get. Whereas ITS is very ubiquitous and present as multiple copies in every organism, okay, as tandem repeats, because ITS is part of that ribosome. Okay, so ribosomes make proteins. Ribosomes is a crucial structure, okay, for organisms. Doesn't matter if it's mushrooms or people. That's why there's so much ITS in things. But for Cantharellus, ITS was difficult to procure because of technical difficulties with sequencing itself. Okay, not because there was little material in the tissue. But now, I guess Steve figured out how to sequence Cantharellus with ITS, so he figured out that also ITS2 is the region in ITS that is a good fingerprint, okay, better than ITS1, which is very variable and intraspecifically variable, apparently, and very long also.
and also as present in any given specimen as ITS1?
I'm sorry, say that again.
Also present as prevalently as ITS1,
Yeah, well, I mean, ITS consists of three parts. It's ITS1, it's 5.8S region in the middle, which is always conserved, 160 base pairs, and then that is followed immediately by ITS2. So it's a continuous string of characters. There are no breaks. There's just no breaks in DNA. Okay, you have a chromosome, point A to point B, millions or billions of base pairs. Okay, that's it. So ITS is just a region.
Gotcha.
And it's identified again, structurally there are three parts to it, but
You're looking at one,
contiguous string of characters.
1, 2, or 3, and all of those 1, 2, or 3s of ITS are, everywhere within the DNA.
Yes, they're everywhere, and you obtain the whole sequence usually as a in one sweep. You have a primer before ITS1, and you have a primer beyond ITS2, and that's how you get the whole thing. But again, with Cantharellus, getting ITS in general has been very, very difficult. That's why if you go back to some of the emails, with Steve, he said, do not send me Llyophyllum, do not send me Cantharellus. I cannot sequence them. But now he can.
And that was the difficulty, right? Because people were only looking at ITS1 and not necessarily ITS2.
No, they were looking at
Right.
everything, but it was just tough to get it to begin with. And then when they could get the whole sequence, they found out that for the same species, ICS1 was not reliable. Just like it is the case with Lexinum, that it's not reliable because it's very variable within a single species. There are like deletions and insertions and other stuff going on. I don't quite understand it, but that's pretty much, in a nutshell, the story. So instead of looking at ITS1, they figured out that ITS2, which is more homogenized, or I should say homogenized, but it's more conserved structurally, okay? It's easier to deal with, and it's still a good barcode.
Cool.
Somehow
Thank
it can
you.
still resolve individual taxa within, Cantharellus to the same
I
extent
appreciate
as Step1 and
you.
RPE2.
I appreciate you talking that out because it's always confusing to me.
It is confusing, and I don't mean to confuse people, but if you don't say anything, we don't get anywhere, right?
Yeah, agreed.
Right. So I mean, if you want to sequence chanterelles, Steve probably would be a good source, for that, because I'm not sure how well you can sequence chanterelles with Sanger, and that's a question for somebody like Linus. Maybe he knows. But in this case, we don't have to sequence these things, because morphologically we can separate Cinnabarinus from corallinus, by two main traits, namely the gills and the presence of these white spots on the stipe for corallinus.
And when you're talking gills, are you talking gill attachment here?
Now I'm just
Or
talking the
spacing?
folds themselves, and that they're shallow and they're also more blunt in corallinus as opposed to Cinnabarinus. So what I'm saying is that in Cinnabarinus, they look more like gills, and they're also thinner and sharper edged.
Whereas this looks more like a chanterelle, right? Chanterelle gills.
Yeah. Yeah, it's more blunt and shallow. Yeah. And there also might be a difference in the spore size that I have to check. If I can probably send you that paper if you don't have it, by Rachel Sweeney. I think I was able to get a copy from somebody.
Yeah, I have it. I've read it.
I might have sent it to you as well before. I don't remember. So
Yeah, my internet dropped out and I just managed to get it back.
that's what we figured, Dave. Welcome
Yeah.
back.
Yeah. So I'm still a little bit dubious on these supposed morphological differences between corallinus and Cinnabarinus, but these do have the white areas on the bottom parts of the stalks. At least most of them do. But this particular collection was not sequenced, but two other ones were. From the exact same spot on my property. We're talking 2 square meters.
Yeah, that's
They come up every year in this same spot in association probably with shagbark hickory, although there's an apple tree there also. But my guess is the hickory has more to do with their being there.
it is interesting that most of these new species of chanterelles that transpired in the last, I would say 5 years, came from southern United States, the type collections. So people are thinking, oh, they only find them, in Texas and Tennessee and Louisiana or whatnot. But apparently they creep up all the way, to the northeast. So do not look at where the type came from. You have to deal with the mushroom itself and be open-minded. So we have both Cinnabarinus and corallinus here in the northeast. That's my understanding.
I believe that there's another collection of cinnabar chanterelles that I sent to Steve that were sequenced, and it's not matching corallinus, and it wasn't really the sequence that was he put up didn't really seem to match very well with anything. But corallinus was on the list of matches, but way down. I mean, very poor matches, like in the 80s.
Yeah, you know why? I've looked at chanterelles before, and I might have written a number of comments on the Mo. From what I remember, there is no ITS sequence from the type collection of Cinnabarinus. So by the process of exclusion, I think Steve said that he sequenced pretty much everything that is to know about Cantharellus in the United States. I think he was able to identify a couple of more cryptic species, but he said everything else, where you know what it is from ITS. So if your pink chanterelle, the sequence of it doesn't match corallinus, by default it must be Cinnabarinus.
That is what I concluded, that it was most likely Cinnabarinus. And those other ones were from Ricketts Glen, which is not that far from where I live. And the habitat is not all that different. It's just that there's a lot more oak, and the woods are older also. There's some like old oaks and hickories in that other spot. So I think there's a few other chanterelles I sent as well, but I don't know what happened when I was. Did you look at them
we
already?
just went to that first one, and this is the second one that you sent.
Oh, okay. Yeah. There's a couple of what we used to call Cibarius, years back, which is.
Well, that's a mess. Cibarius and Elenensis are the ones that are in the same clade, and then we have 4 more species that are Deceptivus. That's a different clade altogether. Phasmatis, and 2 more that I forget.
Yeah, but what I've got Analensis is one of the 2.
Real quick, guys.
Sent.
Sue, can we start sharing your screen?
Oh yes.
Oh, did we lose it?
We lost it. Yeah.
Oh, okay, right. Yeah. There's one that seems to be maybe a somewhat uncommon species. Anyway, there's 2 of them here. One of them is Analensis, I think, and the other one is
Can you everybody see it now?
Yes.
Oh, there's Analensis. Okay, yeah, no, I'm sorry, I got them mixed up. So this is the one that I think.
That's Analensis.
Yeah, I found this one. There's a spot where I used to get it every year. It's posted now. It's no trespassing, so I haven't been there for a while.
And again, the ID is from ITS or DNA. What's the observation number? 139,484. 139.
484. 139,484. Yep. So the sequence is probably there, I'm pretty sure.
Yeah, I'm trying to just basically look here on my computer because I'm.
Yeah, I don't think I blasted any of these. I probably did, and I just forgot.
The sequence is there, and it's a short one, which means it's ITS2.
Oh, okay. I see.
Yeah.
Oh, that's what you were just discussing. I just came in on the end of that discussion, but I think I got the gist of it. Yeah. Okay.
Yeah, I mean, all of the ITS sequences that Steve produces for Cantharellus are going to be short because they're only going to be the ITS2 region of ITS, which is typically like.
I don't want you to have to repeat yourself a whole lot, but what is the problem with ITS1? Is it just that it's so really conserved in Cantharellus?
No, it's that way. It's very variable, and I think there's also there's also a challenge of getting an entire ITS sequence, even with modern methodologies. There's some technical problem. So they were able to, they're able to design primers that begin in the middle of the ITS region, so you get your forward read from the middle of that region or locus, I should say, and then the reverse read goes from LSU into
from LSU
into 5.8S. Yeah.
Okay, that's, you don't have to explain anymore. I get it. I get it. So the ITS one is just, I guess it has like a lot of indels and stuff or whatever.
Yeah, exactly. So when you're trying to align these things, it's just a mess, even within a single species. Just like in Lexinum, I said, it's the same kind of story. Similar.
Just like in Lexinum. Okay, very good. Thanks. You don't need to say anymore because
I'm not going to say anything. No. But I am.
I will ask one more question because Dave, I don't understand this stuff pretty much at all. But starting at 2, does that mean you get more like inside out representation? So, if you start at the middle as opposed to the first third, are you getting more matching on the back half of that middle? If that makes any sense.
You just get better. What Igor's saying is you just get a better representation of the sequence when you only get ITS2, and you can predict where that starts because there's some sort of little region in between 1 and 2 that's either always the same or it's
Yes,
one of
it's
the small set
five
of.
point eight. It's
Gotcha.
5.8
Okay.
s, and it's of a very fixed length, and the points where it begins and ends are well identified, so you can
Right.
clearly
Yeah.
understand. You can clearly see where your regions come in just by looking at the sequence, because you just type in those little primer areas that people use for sequencing, and you can identify them. Anyway, so that's the gist of it. You can annotate your sequence very clearly, easily, by using these short um, uh, uh, um, strings of characters that herald the beginning and the end of a region in any ITS sequence. That's the gist of it. And here I'm looking at the blast of Dave's sequence. That, yes, it's C.enelensis, and a lot of it came from, I think, earlier papers on Cantharellus. So
All right. The last one I think I have from Gabe is the Cantharellus septentrionalis. Dave, are you still with us?
Oh, I'm sorry. I forgot to unmute myself. Yeah, these seem to have more of a pale stipe, although there are a few different collections here. I probably should not have. I should have probably created separate observations for the 2 or 3 different collections here, but they were all made from the same area in this pretty much identical habitat, which was not the same area as the Analensis. So I'm guessing these are all the same. But at any rate, at least one of these little groups of chanterelles is this species here,
septentrionalis,
septentrionalis. Yeah.
which means northern in Latin or whatever language it
Oh,
is.
okay.
So here's the story about septentrionalis. It's a Smith species, right? So for many, many, many years, decades, it was one of these forgotten species. And then somebody described a species called Altipes. I think it must have been Sweeney or somebody who was working on the chanterelles, about maybe 7, 8, 10 years ago. So it turned out when they finally got to the type collections of Smith's chanterelles, this name came out to be as the older name for Altipes, and now it basically is the name for the species, and it's a fairly easily recognizable species. It has a very long stipe and a small cap. So Altipes means long stipe or long foot, I think. So the ratio of the stipe length to the cap width is going to be a large number.
Yeah, and that's
Yeah,
what we
well-
see here. Yep. Oh, okay, great.
And I think they also have very, pallid stipes, if I recall correctly, and, contrasting the darker caps. So all these traits
right.
taken together, septentrionalis apparently is a fairly easy species to ID in the in subgenus or section Cantharellus.
Yeah, these are, they look quite unique, but you can easily tell them that they're a chanterelle, right? With the gills, white stipe, tiny cap. I've been using cap Q as a thing recently. And yeah, that's a big Q, right?
We keep seeing using the word gills for the chanterelles, but they're false gills, not true
They're
gills.
false gills, yeah, but we still call
Yeah,
them gills
false
because
gills.
Yes,
Yep.
I'm just saying for novices who might be on,
Yep.
it's a little misleading to them.
yeah,
Good
they're
point.
false
We
gills,
should just
they're
call it
false.
the Hymenium, probably.
Yeah,
Oh.
Yeah.
ridged Hymenium, yeah, something like that.
Folds.
Yeah,
ridges.
folds.
Yeah, they're false gills. They're false gills. Or yeah, false. I call them ridges myself, but
Yeah, they
definitely
are.
I don't think I want to word just gills by themselves.
The Hymenium is made up of the same context as what composes the rest of the mushroom. So it's not a separate layer of context as would be the case in true gills.
Right. That's what mushroom expert says, is that two different things structurally.
So do all chanterelles smell like peaches or apricots? Apricots, right?
That's what they
The ones
say.
that I find do, including the Flavolataridius.
Yes, that one definitely smells like
Yeah,
apricots.
these do as well. They all are excellent edibles. I've been eating these for years. These tend to be a little bit small, but sometimes you can find enough of them to like a side dish or something, or to add to a meal.
Yeah, I don't think I've seen Altipes or Septentrionalis in New Jersey, but I think we get plenty of Deceptivus and Phasmatis mostly, which is a very difficult clade genetically to separate species from. And Steve basically pretty much said the same thing, that he's still struggling to separate species in that little clade containing those 4 species. Everything else is easy genetically. Unambiguous.
When y'all eat these, are the stipes nice as well, or are they chewy?
Oh, the whole thing is good.
Yeah.
Nice.
The whole mushroom's really good. Yeah, all of the chanterelles are like that.
In terms of the texture, it's very homogeneous. It's
Yes.
there's like no distinction between the cap flesh and the stipe flesh. Maybe the tip, maybe like the bottom gets a little bit tougher, where the ground is and you cut it off, but the rest of it, middle at least up, is the same.
I think that's all you had, Dave, for tonight.
I believe that's all I sent. Yeah, there were 6 things actually, but these are things that I just got results on these, so I thought it would be interesting.
Yes, indeed.
Mm-hmm.
Does anybody else have anything to share? Okay, well, I have one mystery one, and I'm sure it's still going to be a mystery. And someone emailed me this. Notice the bad photography in that we only have the cap without any underside. Does anybody want to guess what it might be?
Yes, I want to guess.
Okay.
I would guess
I
also.
think this is Oudemansiella.
I didn't quite get that.
Oudemansiella, Xerula.
Oh, Xerula. Okay, yeah. I was thinking
can
of
see that.
Hymenopellis, yes.
I was going to say oyster mushroom.
This is the only photo.
When was it found?
It looks like the date is September 29th.
Okay,
Civil year
Oudemansiella
2018.
makes more sense.
2018.
Yeah.
Yes, he's clearing up his back photographs, so he wants to put names to things. But
Yep. The dark oyster mushrooms usually don't show up until pretty late, end of October, November.
And I'll say it looks like central stipe just based off of the cap photo, right?
Okay.
I just see a lot of these wrinkly, these
wrinkly
nice,
caps, yes.
wrinkle caps, and they're kind of radial wrinkles. I mean,
Yeah, the wrinkles fit. Yep. Yep.
So which of the Xerulas do we think that this was going to be? We're not going to go to genus species.
I think we call them Oudemansiella, but there's also Xerula and Hymenopellis as competing generic names.
Yeah,
but I think Oudemansiella is the one that is accepted now commonly.
I've seen a bunch of these posted as Xerula recently. So I don't know.
There's still a difference of opinion.
Yeah, yeah.
The there are other if you want to put this in Oudemansiella, then it's going to be in the same genus as other species that produce mushrooms that look absolutely nothing like these, and
Mhm.
and the habitat is way different. And
Mostly
so
on wood, right?
I think
Mostly
there's a
on wood, right?
well, on wood, yes, but these subtropical things, they call them like porcelain caps or something. They're slimy, they're small, they have rings on the stalk, so I guess they have a
Mhm.
partial veil to begin with.
Yep.
Yeah. They just look completely different, and they grow on like sticks, like small sticks. These grow on buried wood.
Yeah, the only thing I would think that this is strange for Xerula type mushroom is I don't see the stipe.
I was
And
about
those
to
things
say
are
the same
long
thing. You just stole
stipes.
it from my mouth too. I mean, why would somebody remove a stipe, which is like a significant portion of the mushroom? It's very long and slender. Why would you
Yes.
like
I have
completely
an
like cut
answer.
it
I
off?
have a potential answer for that because they broke the stipe and they were embarrassed to show it. Because that's what always happens unless you're really careful with
And
Well, he was only
excavating
interested in
and
seeing
also
it.
and also why is a fern here? I mean, obviously maybe he found it with a fern, but
For contrast
That's
purposes,
garnish.
maybe.
garnish.
Garnish, yeah. You know what, Sue, if you know who that person is, just, tell them next time if they have more than one mushroom to flip the other one upside
Yeah,
down
I
so we
know
can see
him.
the
I
gills.
keep telling him that he's just clearing out old photographs. He knows better now after 6 or 7 years.
There is a brown capped Oudemansiella, and I believe it stains brown on the gills also. And I think it has Bruni or
Yes, it has a long name.
a
I
long
know what you're
name
talking
with
about.
Bruni in it. Yeah.
Yeah, I'm gonna find it. Yeah. We don't see it very often. I think I only saw like once or twice in the last 15 years.
It was at one of the big forays I was at. I think, it was at Sequinota. Somebody found it at Sequinota, the
Yeah,
brown one.
yeah, right. I think you're right, but I still keep referring to that specimen that I saw last time in Wawayanda from 2017. So that was like 8 years ago, almost.
Yeah, but that whole Furfuracea group, I mean, they can present with brownish caps, especially in dry times.
Yeah, these could just be Furfuracea. But they're kind of dark. These two are kind of dark.
Yeah.
Assuming that they are Oudemansiella. I mean,
Yeah,
that looks
assuming
like
that
a
they
pretty
had,
good guess, actually.
assuming that the telltale sign of an Oudemansiella is there, the taproot, right?
Yeah, the name that they was looking for, the one that stains brown prominently on a stipe, is called rubrobrunescens.
So it
Or the
doesn't
ones,
stain red?
Reddish
pardon?
brown.
Okay.
Yeah, reddish brown. Yeah, and you can see here the caps. The left cap. You can see the discoloration, kind of reddish on the left side of that lobe.
Yeah,
Yeah.
vertically all
Oh,
the
I
way on the
see
left.
that. Little stripes. Oh,
Yeah.
Yeah.
interesting. Yeah.
Okay. Well,
I
thanks
think
for
you got
doing
that
the
one,
tree
Igor.
frame one.
Good job.
Yeah, well, Oudemansiellas are, as a genus, are fairly easy to identify just looking at the caps alone. You're gonna see these wrinkles all the time.
Well, I thought oyster mushrooms because I thought maybe they were recently found. Because you find these dark Pleurotus in the winter. But it's not it's not that, especially since these were found in September. They're not the Pleurotus you would find in September would be very pale. Would probably be what's that summer one?
Pulmonarius or something.
Pulmonarius, yeah, it would be that probably. These dark Ostreatus, they don't show up till later in the fall and into the winter.
Now, is that like a different variety, or they just get darker because of the weather, or what's the deal here?
I think Australis is just naturally a somewhat darker capped mushroom, gray to brown, light brown.
Gray, blue.
I think in the winter they're getting more sun.
Do they get more melatonin going, huh?
Yep. Because there's like I see two patches routinely. One is on a tree closer to the fence line, and the other one's out on a stump that's in that direct sunlight. And I gotta assume they're probably the same. They start fruiting at almost the same times. And the one out on the stump, always darker.
Yeah, after catching some rays, huh?
Probably the ones you find out in the woods too are probably because there's no leaves on the trees.
It's interesting because, who's been doing the DNA on Pleurotus? Everyone pretty much feels confident knowing what a Pleurotus is, what an oyster mushroom is. So is there any work actually being done on these things?
I would guess yes, because the names Pleurotus populinus, Pleurotus pulmonarius, and Pleurotus austratus are used typically with a fair amount of confidence applied, and they seem to be seasonal. Populinus is like spring, like May, April, May. Pulmonarius is summer, and austratus tends to be in the fall and the winter. So I'm guessing they're pretty well understood. There's a few other species as well, but they're different. like Levis. Those are different though.
Yeah.
They have like hairy stalks.
Oh yeah, Dryanus and Levis. That's a different story. I've gone down that road a couple times, but yeah, I see what you're saying.
Yeah, there's this guy on Mushroom Observer from Vermont. If you post something and say it's Dryanus, he will just automatically come out and tell you you're wrong. And I don't know I don't know. I mean, according to him, there's like no such thing as Dryanus practically, but I have to wonder about that a little bit.
I think he's on INET as well. I forget his name, but yeah, there's he's got a program going. Anytime somebody posts one of these, he's got a response to it.
Yeah, he's on this like crusade for Levis or something, so
Yeah.
it has to be that. So it is true though that a study was done on those, and an awful lot of what had been assumed to be Dryanus is now believed to be correctly called Levis. I do know that.
Yeah, and I agree with his, crusade, as you put it. It's a good word, but that doesn't mean that Dryanus isn't out here. Dryanus.
Yeah, he seems to be a little bit quick with the Levis. As soon as you post it, like the next day you'll see.
Is the problem where the dividing line, like north and south or something?
I don't know. There might be a.
He's got a big post one, and you'll get the answer, right? Because he gives you it's a whole long thing.
Oh yeah. You get a long answer. I think maybe Dryanus has an actual partial veil it's kind of fleeting, and sometimes it leaves a ring.
Because I found one that I
so does Levis though
had from last year, and I thought I had looked up and I had found, I think, the name Dryanus first, and then we were like, no, it's Levis.
Oh, did you put it online and it turned it up and everybody said it was Levis?
No. We said it was Levis. I put it on.
We said it
as a
was.
leaf.
Yeah. Well, we're part of the crusade now, I guess. Here.
Mm.
So.
Man, I got self-censored by Taxonomy Tuesday. Look at that.
Honestly, I think what it comes down to is the cap and the yellowing on the cap.
The yellowing on the cap. That's right. That's
Yeah.
one thing that supposedly points towards Levis.
Yeah, mine definitely had that.
So according to Google, the way to pronounce Dryanus is Dryanus, not Dryanus. Yeah, well, that's what they say in Latin. You know, I basically like,
Dryanus actually sounds a little better.
yeah, Dryanus.
So
Yeah,
not Dryanus. Okay.
Dryanus. So that's the name stands.
I mean, they keep trying to pass off that Uranus thing too.
Yes, yeah.
But, you know, I know every fifth grader in America says it one way and one way only.
Yep. Well, in this case, you know, you cannot